This is my second time at this summer school, and I thought it's important to educate the next generation of scientists, and I think this is a really good venue for doing that. And so this is slightly different than other meetings in that, you do get to talk about your research, but it's in a much broader context that you're able to use bits and pieces from various projects within the lab and just put it into a much broader context to explain concepts to students rather than going in detail on a specific project. I talked about the importance of using a variety of different molecular profiling platforms to understand functions of proteins that have not previously been described. And specifically, my group is interested in understanding non-catalytic functions of a protein group that is known to carry out a catalytic reaction. So these are kinases. And a couple of years ago, we found that this kinase called AKT, which is a critical player in oncogenesis, is able to promote cancer cell survival through functions that do not require its catalytic activity. But there is no defined path in terms of how you study these novel functions, and it's very difficult because with an enzyme, you can simply do a biochemical assay to read out its activity, but with some of the functions that we've identified now, it's a lot more complicated. But I think what we're doing is trying to provide proof of principle using this example, and to say that you can use a variety of different omics platforms. For example, what we've done is phosphoproteomics, transcriptomics, as well as low-resolution metabolomics through magnetic resonance spectroscopy. And then what you do is you do a variety of different models. You bring together the data from the different platforms, and you look for what's common in all the platforms and across all the models. And that hopefully reduces the number of hits that you get, and then you can take those on and study them further. Because obviously the challenge is that whenever you do these large omics experiments, you end up with a lot of data. And so how you go through it depends on your approach. And the approach here is, let's just do it in tons of different cells and then see what the common denominator is. AKT is a key player in cancer. It's a key component of the PA3 kinase signaling pathway, which is arguably altered in all human cancers. And for that reason, it's being pursued as a therapeutic target for a number of years. Now, there is a disconnect between the frequency of PA3 kinase activating lesions in cancer and drugs that target the pathway being effective at killing these cells. So we basically want to understand why that is. And I think that part of the explanation is that in terms of specifically targeting AKT, is that a lot of the drugs that have been tested in this context only capture the catalytic functions of AKT. So these are drugs that turn off its ability to phosphorylate protein substrates. But as I said, it seems that AKT can do things other than phosphorylate substrates. So I think what we're trying to do here is first more fully understand how AKT promotes tumor growth. But at the same time, I think while doing that, we will identify novel therapeutic targets that then we can use to develop new drugs that more hopefully effectively target AKT-dependent cancers. It's slightly different than most other meetings where you just hear people talk about their latest piece of research. Here you have the opportunity to talk about many things and just put it into a broader context, because I think the purpose of this is to educate scientists in training. And a lot of these people are first-year students, for example. They may not necessarily have all the necessary background to fully process the kind of information that you get at more specialized meetings. So I think this meeting stands out in that it provides you with the opportunity to talk about many things. It attracts some very key players, particularly in Europe. But of course, there's also people from the U.S. And yes, I myself have already talked to a couple of people about potential collaborations, and yeah, it's good. It's always good. 20 years ago, science had a completely different model, and it was every team for itself. And in the last several years, I guess the community has realized that in order to make real progress, you need to work collectively, and not just on a national scale, but on an international scale. So, we're definitely seeing science becoming a global enterprise. And the thing that facilitates this is these types of meetings where you start finding people with common interests and talking about specific projects where there might be a synergy between the two groups. And so this and other meetings are very good opportunities to do that. Well, that's the hope, and I think that there is, it needs to be incentivized, and I think there are some incentives now because a lot of funding streams are really aimed at funding, collaborative work, and I think that's one step in the right direction. This is particularly challenging for new investigators such as myself because we rely on productivity to be able to go up through the tenure review process, and so and there's this and there has to be some institutional support in this sense. What I mean by that is, if you have, say, 5 different collaborators, and you're working on a couple of different projects with these people, you may end up being one of many authors on a paper, and scientific publications remain our currency in science, and therefore I will be evaluated on that. Now, in the old days, it would be seen as, oh well, you only did a small fraction of the work, therefore you should not be rewarded as much. But I think now people are starting to realize that this is the only way to advance science. And so when you get evaluated, you have to be evaluated in that context. And as I said, I think a lot of this depends on the institution where you work at, because they're the ones who are going to evaluate you, and they have to take into consideration the fact that you're doing this work because you really want to make progress, and this is the best way forward. Science is very slow, and it's very easy to get disappointed. You just have to keep trying. And I guess the other thing that I've faced many times during my career is I've encountered questions in which the answers seem to go against what most people think. And so when you actually get that, and if you're sure that your data is strong, then just try not to think about what other people think, even if it's everybody else that thinks differently. If you're sure of your data, just push for it. That's how you bring about change. And a lot of times you have to change dogma. So just don't be afraid of doing that.